Welcome to AI for Good, the leading action-oriented, global and inclusive United Nations platform on AI. Organized by ITU, in partnership with 40 UN sister organizations, and co-convened with Switzerland. The goal of AI for Good is to identify practical applications of AI to advance the United Nations Sustainable Development Goals and scale those solutions for global impact. In today's session, we're counting on you to use the live video wall feature to ask questions and post comments to help create an engaging discussion. We encourage you to stay until the end to chat, connect, ask questions, and network with our distinguished panelists and world-class AI experts in the neural network. It is now time to kick off the session and welcome our first speaker. The floor is yours.
Thank you, Anna. Hello, everyone. Good morning, good afternoon, good evening from me as well. I am Guilherme Martinez Roura from the International Telecommunication Union, and I would like to welcome you all for joining today's AI for Good event on AI as translator interface to the wisdom of the living body, organized by the ITU. Today's session will delve into how bioelectricity powers living robots like Xenobots, which self-assemble, navigate, self-repair, and even replicate. This fusion of robotics and regenerative biology enables adaptive, self-healing machines with AI decoding bioelectric signals for biomedical and environmental breakthroughs. This session is part of the AI and Robotics Discovery Series, which features expert talks on the state-of-the-art developments in autonomous robotic systems to advance the United Nations Sustainable Development Goals. We have a very distinguished speaker today, but we, as always, we're counting on you, the participants and the audience, to help create a very interactive session. As a reminder, the audience can leave questions in the AI for Good neural network chat room that will be answered during the Q&A section. So without further ado, I would like to introduce our speaker today. His name is Michael Levin, and he is a distinguished professor in the biology department and Vannevar Bush chair at Tufts University. Michael, welcome, and the floor is all yours.
Let's see. All right. Thank you so much for having me here to share some ideas with you. If anybody's interested in following up, more information is at these two websites. What we're going to talk about today is the use of AI and biorobotics as an interface to the wisdom of living material. And in particularly, we have had this framework described by this ancient kind of picture of the world where Adam, the human, is separate from these other animals. They are discrete. There's a certain set of animals. We all know what they are. We know their properties. There are different species and so on. And there are two interesting things about this old piece of art. One of them is that it is on Adam to name the animals. Okay, so in this original story, God couldn't do it. The angels couldn't do it. It was on Adam to name the animals. And I think this is very profound, because as in those traditions, naming something means that you have discovered its true inner nature. And I think this is the challenge for us going forward, is to discover the inner nature of numerous beings that are not in this painting, that are not in this set of existing animals. And we're going to be, much as Adam had to live with them, we are going to be living in this novel world with these new types of beings, and we are going to have to discover how to relate to them. So today I'm going to tell you roughly three things. First, we're going to talk about some limiting assumptions that have held back progress and that we need to overcome if we're going to achieve the kind of flourishing of the humans and other species on Earth. And these assumptions are mechanism, anthropocentrism, and an over-reliance on the trial and error processes of natural evolution. And then I'm going to tell you about bioelectricity as an interface to the plasticity, reprogrammability, and intelligence of life. And then I'm going to talk about biological robots, or biobots, and the use of that platform and AI to address basically three fundamental things. First of all, the unsustainable health spiral, the idea that what we are doing right now is spending most of our resources on an ever-sinking ship of the body. That is, as we get older and older, the interventions needed to keep us alive get more and more expensive and more drastic, and most of the resources are going into developing these kinds of applications that keep us going longer. But of course, in that scenario, each one is harder and harder because the body is consistently degrading. So we are going to try to break that spiral, and as well as have some technology for addressing some environmental challenges and some ethical challenges of living with beings that are not human minds. So let's first talk about the basic idea of where do our bodies actually come from. We all start life as a collection of embryonic cells, and then through an amazing process of self-assembly, we become this incredibly complex anatomy. So this is a cross-section through a human torso, and you see all of the organs are in the right place, the right location, the right size, the right relationship with each other. Where does this information reside? How do these cells know what to do? And you might initially think that it's in the genome. You know, there's a human genome, and many people think that the genome is what sets all of these properties. But we now know how to read genomes, and we know that what the genome actually specifies are the proteins, the tiny molecular hardware present in each of your cells. The genome doesn't directly specify any of this. Any more than the termite genome specifies the structure of the mound that they live in, or the spider genome tells you exactly what the web is going to look like. The resulting construction projects are the process of self-assembly and decision-making and building by cells that are using that genetically specified hardware. So between that layer, between the genetics and the actual outcome, lies a very important process, which is fundamentally intelligent. And that process allows cells to know what to build, to know when to stop, and that allows us to, as far as we understand this process, we can communicate with cells to force them to rebuild, or encourage them actually to rebuild whatever's missing. And as engineers, we would like to understand what else is possible to build. With that same genome, what else could those cells build? And in this field, we think of as an endgame of of an anatomical compiler. The idea is that someday you will be able to sit in front of a computer and draw at the level of anatomy, not molecular properties, but actually the anatomy that you want of the organ, the body, the biobot, plant or animal, whatever it is that you want in whatever shape you want. You'll be able to draw it, and the anatomical compiler will compile that description, that anatomical description, into a set of stimuli that have to be given to cells to get them to build exactly what you want. Now, why do we need this? Because if we had the ability to make cells build whatever we want, all of this, birth defects, traumatic injury, cancer, aging, degenerative disease, all of these things would go away. All of these are fundamentally problems of information processing and the inability for us to tell cells what they should build. Now, this is really critical. This system is not some kind of a 3D printer that will put every cell where it goes. This is really a communications device. And this is the reason we don't yet have something like this, is because we've been focusing on the hardware. But actually what we need is a translator device from our needs as workers in regenerative medicine and bioengineering to the collective intelligence of cells. And so where biomedicine is today and bioengineering are that we are very good at manipulating cells and molecules, so figuring out which genes and proteins interact with which other genes and proteins, but we're really quite a long way away from control of large-scale form and function. If somebody loses a limb, if somebody has a birth defect, and so on, we're quite still a far distance from enabling definitive repair, and that's because biomedicine has been roughly where computer science was in the 1940s and 50s. What you can see here is that she's programming this device by physically rewiring it. And that's where medicine is today. We are, all the most exciting advances have been in genome editing, protein engineering, pathway rewiring. All of these things are about the hardware. And a lot of it has been driven by the assumption that the body is a complex machine with DNA encoding outcomes. But actually, we are now seeing in this new emergent field that ties together studies of diverse intelligence and unconventional cognition with biophysics and regenerative medicine, that what we're really dealing with is a highly reprogrammable agential material. And so I will show you just a few examples. This is a tadpole. It's a tadpole of the frog Xenopus laevis. Here's the mouth, the nostrils, the brain, the gut, and here's the tail. What you'll notice is that this animal doesn't have primary eyes. What we have done is put an eye on its tail, okay? And when we do that, what happens is that the eye forms perfectly well. It puts out an optic nerve. Sometimes that optic nerve synapses onto the spinal cord, sometimes on the gut, sometimes nowhere at all. But if you test these animals in this machine, and we built this machine to test them on visual learning cues, you will find out that these animals can see perfectly well. Now, isn't this remarkable? In one generation, without any further rounds of mutation, selection, an animal with a radically different sensory motor architecture, the eyes are now on the tail and not actually connected to the brain at all, this animal can see immediately, right out of the box. No new adaptation. Why is that? Isn't that remarkable and unexpected? Given the story that we've all studied, that the long eons of mutation and selection are needed to provide integrated system function like this. We can also think about examples like this. All over the world, reliably, these acorns grow up to be oaks with leaves in precisely this shape. And so because it's so reliable and happens so often, we might think that actually the oak genome encodes this particular pattern. This is what it knows how to do. And we would never know that actually the situation is much more plastic if it wasn't for a bioengineer. Now, this is a non-human bioengineer. This is a little wasp. And what this wasp does is lay down some signals that prompt the surrounding cells to build something completely different. These are called galls, and here's an example, and here's another example. Now, look, these are not made by the wasp cells. This is made from the leaf cells of the plant. Now, if we didn't know this was possible, we would have no idea that the genome that naturally builds these flat green things is actually also capable of building something with this remarkable complexity. The process is reliable, but it is also reprogrammable. And other creatures, including human bioengineers and wasps and many other things, are able to take advantage of that and to hack that plasticity for new outcomes. Just think, what could you build, right, out of plant or animal cells if you understood the encoding of shape to the extent that you could make flat green leaf cells build these kinds of things. Okay, and so the reason all this works, the reason this incredible plasticity works, is that we are made of cells that are themselves very competent. So here's a single cell organism. This is called a lacrimaria. It has no brain, no nervous system, no stem cells, just one cell, which is very capable of handling all of its physiological, its metabolic, its behavioral needs in one cell. This is the sort of thing that we're made of. And we, in fact, start life as a single cell, the fertilized egg, and then reliably, slowly and gradually self-assemble into one of these incredibly complex creatures, including a complex metacognitive mind. But all of us are built of this kind of active matter with agendas. Not only are cells highly competent, but in fact, the material that they're made of, the molecular networks themselves, are also have various kinds of protocognitive capacity. For example, they can learn. So the molecular networks inside of cells are capable of 6 different kinds of learning, including Pavlovian conditioning, as you see here, and that these molecular networks are already, the material of which even single cells are made is already quite capable. And in our lab, we're building devices to actually train these networks so that we can take advantage of their memory properties for biomedicine, for drug conditioning and things like that. So your entire body and life in general, all the way from molecular networks up through swarms of organisms and ecosystems, has a kind of nested intelligence, not just nested structure, but actually problem-solving capacity in different spaces, not just in 3-dimensional space, as we're used to thinking about for animals that move around, but all kinds of other spaces, physiological spaces and gene expression spaces and anatomical spaces. And when I say intelligence, what I mean is William James's definition, which is the ability to reach the same goal by different means. Okay, so some degree of competency that when the circumstances change, when there are barriers, when things are different, you can still meet your goals by whatever means are necessary. And let me show you a couple of examples. So we know that the navigation in anatomical space, when you start out as a single cell, and then eventually you have to undergo all kinds of changes to become a human body or a salamander or a tree or a snake, that journey through anatomical space is not hardwired. It is not the same path every single time. And we know this because, for example, if an early embryo, including an early human embryo, is split into pieces, you don't get half bodies. You get perfectly normal monozygotic twins and triplets and so on. Each part knows that the other part is missing. It can reach that same goal by novel steps, such as rebuilding an entire half of the body. Some animals can keep this capacity throughout their lifespan. For example, this is a salamander. These guys, this is an axolotl amphibian that can regenerate its eyes, its jaws, its limbs, its spinal cord, portions of the heart and brain. And if they lose a limb anywhere along this axis, the cells will do exactly what they need to do to build a perfect replica, and then they stop. And this is the most amazing thing about the process, is that they know when to stop. When do they stop? They stop when the correct salamander limb has been completed. So this is a process of anatomical homeostasis. It is deviated from the correct space in the correct location in anatomical space, and then it will work very hard to get back there, and then it stops. In fact, it's even more clever than that. If you take a tail, an amphibian tail, and you graft it onto the side here where the limb normally goes, so you would graft it up here, what happens is that slowly over time, that tail actually remodels to be a limb. And even the cells at the tip of the tail, which are locally fine, they're tail tip cells sitting at the end of a tail, they will slowly remodel into fingers, the structure more appropriate to the large scale pattern. So all of these scenarios are showing you that the cells form a collective intelligence. The individual cells don't know what a finger is or what a tail is or anything like that, but the collective certainly does. It knows what the final shape is supposed to be at the organism level, and it can follow the steps to get there. Now, how does it know what to do? How does it remember what the correct shape is? How does it calculate where to go? Well, if you want to understand how groups of cells think about goals and how to get there, the most obvious example, of course, is the brain. So we're used to animals with brains and nervous systems being able to remember goals and to act in three-dimensional space to implement those goals. So the way it works in the brain is that there is the hardware are the cells of your brain and nervous system. They run an interesting kind of software, which is the electrophysiological dynamics, all of the electrical signals that are moving inside that neural network to process information, driven by ion channel proteins that sit on the surface of these neural cells. And the dynamics of these channels opening and closing so that electrical information can propagate and be computed throughout the network allows signals to be sent to muscles, which move the body in three-dimensional space. And then there's this project that neuroscientists do called neural decoding, where the idea is that if you could just read and decode the electrical activity, you could read out the memories, the goals, the preferences, all of the cognitive content of that being. All of it is encoded in the electrophysiology. Now, that's an amazing architecture, and we can see the beauty of it in the world of animals with brains. But it turns out that actually this system and evolution's use of electricity to process information and to integrate goals across space and time is extremely ancient, because that same scheme works in every cell of your body. So every cell of your body has these same ion channels. Most of them have electrical synapses that bind them together into electrical networks. All of them drive very particular electrical patterns. And instead of telling your muscles what to do to move you through three-dimensional space, what does this system think about? It thinks about shape. It thinks about anatomy, and it issues commands to an embryo cell or to a regenerating organ, in fact, to all cells during your lifetime to keep them away from cancer and towards building healthy organs. It tells all of the cells how to move your body in anatomical space. They remember, they encode literally memories of the large-scale shape of your body. And what we've been developing are tools to do this kind of non-neural decoding, to try to read the electrical mind of your body and to understand what patterns it's going to try to implement, and then to modify those patterns and to convince it to build something else. And this system is extremely ancient. It long predates nerve and muscle and all of that. In fact, bacterial biofilms already have some of these properties. So I want to show you what it looks like. We developed voltage-sensitive fluorescent dye methods to actually look at the electrical properties of living tissue during this kind of decision-making, during anatomical decision-making. This is a frog embryo putting its face together. So this is a time-lapse movie. So if you think about this very old idea of a kind of energetic scaffold that underlies growth and form, we can now directly see it, and we can now make videos of it. This is where the state-of-the-art technology is. So this is one frame from that video, and you can already see that the colors represent the voltage of the individual cells. And long before the face is actually formed, you can see here's where the eye is going to go, here's where the mouth is going to go, here are the placodes for the other organs. That pre-pattern, that memory that tells the cells what a normal frog face looks like is already here. And then, in fact, not only do these electrical memories organize cells into large-scale bodies, into embryonic and adult tissues, but actually they also work at a larger scale between individuals. So here you can see once this embryo here triggers a wave, a calcium wave, and you can see all the other embryos in its vicinity get the message, and the same thing is happening here. It propagates. And so this is what the bioelectrics of individual cells looks like. Okay, and here you can see all the voltage states as they're either leaving the collective or not, and you can see that this also happens between individuals. So it's a multi-scale way to coordinate information across groups, whether that be groups of cells or groups of individuals. So we've also developed ways to rewrite those electrical patterns. We do not use any applied electromagnetics. There are no electrodes, there are no frequencies, there are no magnets. What we do instead is manipulate the interface that cells usually use to control each other's behavior, and that is the ion channels on their surface and the electrical synapses by which they pass electric states to each other. So here's a network of cells, they're all connected, and we can go in and we can manipulate who talks to whom, or what the actual electrical states are that are propagating through. And this is just like what neuroscientists do in the brain, we are doing this in the rest of the body. And when you do this, you can not only read out the pattern memories during the production of different body structures, but we can actually rewrite them, and we can incept false memories into the collective intelligence of the body tissues. Here's an example. Again, here's your tadpole. There's the mouth up here. Here are the eyes. Here's the brain, and this is the gut. What we've done is inject a few cells here with an mRNA that encodes a very particular potassium channel, and we did this in order to recreate that little eye spot that I showed you, that little spot of depolarization that takes place in the electric face, to put it somewhere else. We said if we take that bioelectric state and we introduce it somewhere else in the body, could we tell those cells to build an eye somewhere else? And the answer is yes. Once you provide that information, these cells are happy enough to build an eye. There it is. These eyes have all the right internal structures, so lens, retina, optic nerve, all of these things you can see here. And in fact, something else that's really fun is that if you only inject a few cells, so here we only injected these cells that are blue, they will themselves organize their surrounding cells and recruit them to finish building the eye, because there's not enough of them, and they know there's not enough of them. And so what they do is they recruit the rest of the cells to finish the project, and we didn't inject any of these others, right? So this is a secondary instruction, just like other collective intelligences like ants and termites will recruit members of the team to go move a heavier object. So this ability to respond to stimuli, to have an extremely complicated behavioral response, to build an entire eye from our very simple modular signal that just says build an eye here. Okay, it's like a very high level subroutine call that tells cells what to do. The ability to integrate all of the downstream steps, the individual gene expression, the cell behaviors, all of it, and by the way, to scale to the task at hand, is all properties of the material. Okay, all of this only works not because we're so smart and micromanage the creation of an eye, but because the material itself is extremely competent and we found the interface to it. That same interface can be used for applications such as limb regeneration. So here's a frog. Frogs, unlike salamanders, do not regenerate their limbs as adults. So if they lose a limb, 45 days later, there's nothing. We've developed a bioelectric cocktail that was applied for just 24 hours. And then you can see what happens. The pro-regenerative genes come on, the leg starts growing. Here you've got some toes, you've got a toenail. And in the longest experiment that we have, a 24-hour stimulation leads to a year and a half of normal leg growth, during which time we don't touch it at all. Okay, this is not about micromanaging the cells. This is about communicating on the very first day, communicating to the cells that you should go down the leg building path not the scarring path. And eventually you get a nice a leg that is both touch sensitive and motile. So here now with David Kaplan's lab, and I have to do a disclosure here. There's a company called Morphoceuticals, which has taken this work and trying to now move it into mammals, so that ultimately we can hopefully induce limb and other organ regeneration in human patients. And then the final piece of biomedicine that I want to show you has to do with cancer. So I showed you that individual cells are very competent at individual cell little tiny agendas, right? So very local small goals like hunger levels and things like that, pH and so on. But what happens in development and in evolution is that cells join into networks, and these networks have a much larger cognitive light cone, meaning that the size of the goals towards which they can work becomes bigger. The network can remember larger goals than individual cells can. So while this individual cell is working on its own tiny little metabolic and biochemical goals, the collective here is working on a goal in anatomical space. Individual cells don't know how many fingers as you're supposed to have, but the collective can remember. And what happens is that if they lose it, they will build exactly the right thing, right? So this is the grandiose goal that they're working on, this enormous construction project. And you know it's a goal because if you try to deviate them from it by amputation, for example, they will go back and rebuild. But that process, the ability to work together into a network that can store high-level goals, can break down. Okay, it has a failure mode, and that failure mode is known as cancer. So what you're seeing here is human glioblastoma cells in culture, and what's happened is that they're not more selfish than normal cells. A lot of modeling of cancer cells treats them as less cooperative or more selfish. They're not more selfish; they just have smaller selves. So to the extent that this is an example of collective intelligence, where a higher order self with higher order goals has formed by linking cells into a network, this is a breakdown of that collective intelligence, basically a dissociative disorder. Cancer is basically a dissociative identity disorder of the morphogenetic intelligence of the body. It's again where the boundaries between the cell and the rest of the world shrink down, and no longer does it remember these large scale goals. It's just rolled back to a unicellular ancestor state where all it cares about is itself, and the rest of the body is just environment as far as it's concerned. So that border between self and world has shrunk. So that unusual way of thinking about it invites us to try a different kind of therapeutic modality. Instead of trying to kill those cells with toxic chemotherapy, what if we could do some kind of integration therapy to reconnect them to that network? So this is our model. Here's a tadpole that's been injected with a human oncogene, which makes a tumor. And you can already see that the cells at the site of that oncogene injection have depolarized. So they have a weird voltage pattern different from their neighbors, and they're going to go off and metastasize. Well, what we've shown is that if you inject some ion channel RNA, which forces the cells into proper electrical state and into electrical communication with their neighbors, even though the oncoprotein is blazingly expressed here, it's labeled with red dye. I mean, it's actually everywhere, but look at this location. This is the same animal. Tons of the oncoprotein are here, but there is no tumor, because what actually drives is not the genetic state. It's not the genetic error. The genetic error is actually still here. It's not that. It's the physiological decision-making that are made either by individual cells, which is what's happening here, or by the collective here, right? So here the individual cells are going to crawl off and metastasize. Here there's no tumor because we've reconnected them to the collective. So now you can see that these ideas about thinking about the plasticity, the decision-making, the reprogrammability, and the boundaries between a cognitive agent and the outside world are really very practical. They lead to specific and practical applications. And so now here's what all of this has to do with biorobotics and AI. Engineering over thousands of years has taken place with passive materials. Passive materials, it's up to you to control every piece of the construction. You're going to micromanage it. The good news is that there's not much surprise. They just do the materials just do what you tell them, which is basically to hold their shape. But the bad news is that you lose a lot of flexibility, and you have to control everything. So since then we've developed active matter, computational material, and what we're talking about today, agential material. The thing about agential materials is that when you engineer with them, you have to do it very differently than engineering with passive matter. If you want to make a tower out of Legos is very simple. You have to put every Lego exactly where it goes. But if the tower gets knocked over, that's it. You have to come back and you have to fix it, and you have to know how to fix it. But imagine trying to make a tower out of dogs. You can't do it the same way. You can't just stack them one on top of each other. They won't stay. But if you train them, not only do you get a tower, but you get a self-healing tower, because if it falls off, it will automatically regenerate itself as the dogs go back to their goals. So engineering with agential material is much more about cooperation with the material, communication, and training. It is not about the kind of micromanagement, and this is critical. So let me show you two biological robots made of frog cells. So here you go. These are frog cells. They're basically prospective skin cells from an early frog embryo. They were liberated from the rest of the frog, put together in a little environment. And what they do is they come together. Okay, and they overnight they sort of coalesce, and they make this thing. If you actually look real close, here's a set of them. They just happen to look like this little horse-like shape, but you can see it's moving around. It's interacting with. It'll be a little calcium signaling flash here. There it is. The cells are interacting with each other, and eventually they all come together and they form this. This is we call this a Xenobot, because Xenopus laevis is the name of the frog, and this is a biorobotics platform. They swim along with these little hairs that they wave. They have collective behaviors. They can go in circles. They can go back and forth like this. They have all kinds of interesting computations that are going on inside. There's no brain. There's no nervous system. These are just skin cells. But you can see there's all sorts of interesting computations going. Here's one traversing a maze. So you can see it goes down this arm. It takes the corner without having to bump into the opposite wall. And then here, for some reason, it turns around and goes back where it came from. So they're spontaneously self-motile. They have this kind of behavior that is actually quite rich. And they do some amazing things like kinematic self-replication. For example, if you give them a bunch of skin cells, what they will do is they will collect the skin cells into little bots, little balls here. And each one of these little balls will mature to be the next generation of Xenobots. And guess what they do? They do exactly the same thing. And so you get multiple generations of these bots. So this is a completely emergent behavior. We had absolutely no idea that this was going to happen until we tested it out. And this is something that in our project with Josh Bongard's lab, they did a lot of computational modeling to show us how specific shapes of bots are better at making subsequent generations. Okay, so this is a novel competency. Now, lest you think that this is something specific about frog cells, this ability to regain a completely new body structure, set of behaviors with the same genome, I want to show you this. We call these Anthrobots. These are made of actually human tracheal epithelial cells, adult tissue, so donated by elderly patients during tracheal biopsies. And if you saw this, you would never know that this has a normal human genome. This looked like something we might have found in a pond somewhere, and you might guess that it would have a primitive genome like those kinds of organisms, but actually it's 100% Homo sapiens. There is no genome editing here. There are no weird nanomaterials, no drugs, no synthetic biology circuits. These cells, when taken out of the patient, reboot their lifestyle into this kind of novel creature which swims around, and it too has some amazing properties. For example, if you put it on a bed of human neurons where you made a big scratch, so here's a wound, a neural wound that was made, they assemble into something we've called a superbot cluster. So they sit there, and about 4 days later, if you pick them up, you'll see that what they're doing here you can see this better. What they're doing is they're actually knitting together the two sides of the wound, right? So who would have thought that your skin, your tracheal epithelial cells that sit there quietly in your airway, sort of keeping the mucus going, that given the opportunity, they can reassemble into an entirely novel kind of structure and function and have reparative properties. So this plasticity, okay, the theme of this entire talk is plasticity and reprogrammability. When you engineer with these things, we don't try to micromanage them. We don't try to program in these kinds of properties. They already know how to do a remarkable number of things. Our goal is to behavior shape them toward useful applications. So in the end, here's what I think future medicine is going to look like. Here's where everything is today. These kind of bottom-up interventions, okay, that are being developed today. But we're going to complement all of that with top-down interventions having to do with training cells, agential implants such as Anthrobots that could be made of patient-specific cells. They could be made of your own cells, so they don't need any kind of immunosuppression. They could go in your body and execute repair. And of course, all sorts of large-scale repair events using a class of drugs we call morphoceuticals, which again are not trying to micromanage gene expression or trying to hold specific pathways, but are providing information, actual guidance information to the collective intelligence of cells that says build a limb, build an eye, repair various structures, and so on. So I think in the future, medicine is going to look a lot more like a kind of somatic psychiatry than it's going to look like chemistry, because the goal is to understand the competencies, the goals, the degree of learning capacity, and other cognitive properties of the material with which they're building. And in order to do that, AI is an essential tool, because in order to crack that code and to be able to communicate with these tissues, okay, so here are some cells with some neurons. This is all the bioelectrical activity that's happening here. In order to crack that code and understand how to specify the different outcomes that we want across birth defects, across regeneration, across cancer, we are going to need to have a translator interface. We're going to need to crack that code. And you can envision a set of applications where on devices with which we can communicate, perhaps with language models or in other ways, they're going to be the translator between our goals and the goals and the plasticity and the reprogrammability of the collective intelligence. And the final thing to realize is that as we understand the plasticity of the material, then all of these kind of much wider embodiments of intelligence become possible. Some of these already exist, so cyborgs, humans with various technological appliances. But every combination in the future, every combination of evolved material, engineered components, and software, and together with the laws of computation and mathematics, are providing a very wide range of bodies and minds, of which Darwin's variety of life on Earth, the endless forms most beautiful, as he called it, are just a tiny little corner of that space of possible beings. So cyborgs, hybrids, chimeras of all kinds are going to be with us, and we need to understand these diverse embodied minds in order to have an ethical synthbiosis with them as the whole biosphere flourishes into the future. So I will just stop here and just summarize by saying that intelligence is not about brains. It is extremely ancient and fundamental. It is baked into the agential material of life and actually well beyond living material as well. Regenerative medicine applications will come from learning to communicate, not micromanage the competencies of cells in the body. And bioelectricity is a very powerful interface to that cognitive capacity of the body. And it will lead to regenerative medicine of birth defects, of cancer, and so on. And then the benefits of biorobotics are several. First of all, of course, useful living machines for environmental remediation and sensing, personalized interventions for in-the-body healing, helping us learn to crack the morphogenetic code, and really helping us to expand this anthropocentric view of life and mind that goes beyond the standard evolved human case. And AI will be an extremely valuable tool for this emerging field of diverse intelligence to help us recognize, create, and ethically relate to that space of bodies and minds. And I'll stop here after just thanking. So here are the postdocs and graduate students and our collaborators who've done the work. I want to thank our funders who over decades have supported this work. Very important disclosure. There are 3 companies that are taking some of this work into applications in biomedicine and in the environment and so on. So we hope that these will be practical things that help everything in the ecosystem basically. And as always, we thank the actual model systems because they're the ones who teach us everything. So thank you. I'll take questions.
Well, thank you, Michael, for such a fascinating presentation. Now we would really like to hear from the audience. So if you have any questions, please type them into the AI for Good Neural Network. And in the meantime, Michael, I wanted to ask you a few questions while we are waiting for questions from the audience. So you mentioned that Xenobots can self-repair and even replicate. So what are the ethical considerations you keep in mind when creating these self-replicating biological robots?
Yeah. So with respect to that specific technology, if the risk profile is extremely low, because these things, when I say they replicate only in a very defined set of conditions. You have to provide them with the specific skin cells of which to build. And Xenobots only live less than a week. So they biodegrade naturally after around 7 to 8 days. And so frog skin cells are being shed into lakes all over the world and ponds all the time. And so this is by itself, this is a very low risk technology. Now there are related technologies having to do with transgenic bacteria, transgenic viruses, various kinds of animal lineages that can grow exponentially and that live for a long period of time, and that's a much different it's a much different scenario. But I think in general the question of ethics is really critical, and I think that it's very important to look at it from two perspectives. One is the risk factor, which is okay, what are all the ways that this could go wrong, and that's absolutely important. But let's not forget there's a flip side to this, which is the opportunity cost of missing out on technologies that are here to solve pressing problems. So there's a widespread kind of way of thinking about these things where people implicitly assume that everything is fine, and that what we want is for these new advances not to make things worse, right? We want science and technology not to negatively impact us. But let's be clear, things are absolutely not fine. We have incredibly pressing needs, both on the biomedical front with suffering all over the world in terms of human and capability and animal suffering as well, and environmental. We're facing massive environmental challenges. So the opportunity cost must be balanced with the risk assessment. And so this is something that we do all the time. We think about what are the pressing problems that this could solve and what is the safest way to do it.
Okay, well, that's great. So we have one of the members of the audience asking why were frog cells used specifically in that particular case? Like that's a broader question, but like how you select the different types of cells per different types of applications.
Yeah. There's nothing inherently special about frog cells, as you just saw. I showed it to you using human cells as well. But frog cells have some very nice properties. First of all, they don't require sterility. They don't require any kind of a special incubator, or they live at room temperature on your table in a petri dish. They have very minimal requirements. And for out of the body applications, right? So for sensing, for remediation, for cleanup, for all these kinds of things, you want something that's going to be able to live for a few days anyway in the external environment. Frog cells are ideal for that. And of course, frog as a model of embryonic development, frogs have been used for many decades, because that whole process happens right in front of you. It's not something that, for example, in mice, it's inside the mother mouse, so you can't really see any of it. Here, everything is right there. The whole thing self-assembles, in a visible way, and it just has some very nice properties as a biorobotics platform.
Okay. And I wanted to ask you as well about how, so like all these new materials are potentially going to change how we look into different robotics applications, different materials that were being used for robotics applications. What do you think are going to be the major breakthroughs in bio-hybrid robotics?
Yeah, I think one of the most enabling conceptual breakthroughs is this idea of a continuum between evolved and designed systems. So the traditional way of looking at this is we've got living things and then we've got machines. And I think this is an ancient kind of a pre-scientific categorization that has been really limiting for advances. So what we have now in the field are all kinds of active matter, minimal dynamical chemical systems, such as droplets, and just all kinds of things that are not easily fitting into either one of those categories. And the future belongs to dissolving these binary categories towards a much more flexible description of what is it that your material is capable of. What competencies, what agendas might it have, all the way from completely passive matter through active matter, through the kinds of things that biological systems do. But we call life those things that are very good at scaling up the competencies of their parts. right? And so that's a continuum, that's a spectrum. And so the future of the robotics of the future are going to be an extremely diverse combination of passive materials where we engineer them and we know what they're going to do and they do exactly what we want them to do, and all kinds of other active matter systems, be they chemical, be they mechanical, be they electrical or optical, many other technologies, where you're not just telling the material what to do, you're taking advantage of the computational and the agential properties of the material. So the future robotics is going to be extremely diverse along that continuum.
And you see like this really paradigm shift as a huge potential for reaching the sustainable development goals, for example, in medical applications, prosthetics. How do you see, how far are we from reaching this stage in which we see all these innovations in our daily lives?
Yeah. So I can't obviously give you precise timelines because this is research and this depends massively not only on how the science goes, but how the funding goes, how the the safety testing goes, and all of that. But I will say that many of these things are already in preclinical or clinical testing. So this is not some kind of science fiction thing that will happen long after we're gone. This is we are going to see these things in our lifetime, I think. And many of these are already being commercialized, meaning that people are exploring useful ways to deploy these things for radical, for example, in the case of regenerative medicine. I mean, as I said in the beginning, this idea of patching up a sinking ship, that you get older and older, and we're doing these heroic interventions, increasingly more expensive interventions to just try to keep going. This is not what anybody wants, right? You want an increased health span where the tissues are regenerating themselves just like they did when you were young. And I think this is possible. There are many people, it's not just my lab, there are many people working on this. And ultimately, it's going to be completely transformative, not only for individual human experience on Earth, but actually for the economics of societies, because we're going to be able to break this unsustainable spiral of increasingly more costly interventions into old age and facilitate a definitive regenerative medicine that regenerates healthy tissues as you go along. So this will be have massive implications. And the same thing, I think, for environment and for engineering, for exploration, space, for all those things.
What do you think are the main challenges that you need to overcome, to really push these boundaries and accelerate progress towards these innovations?
Really, I guess three things. One is conceptual. So these binary categories that have been around forever between living things and machines, between hardware and software, between what people think of as real intelligence and these kind of problem-solving capacities that people have a very hard time thinking about them as real cognition. All of these distinctions are a massive brake on progress, and there's been a lot of work done out of a number of both labs and also philosophers over the recent years in the field of diverse intelligence to try to break that down and pull engineering and biomedicine out of that ancient framework. So that, I think, is very important. Number two is specific kinds of big data. So laboratory automation that will allow massive amounts of data on the behavioral capacities, the bioelectric states, the biomechanical states, all of the experiments that need to be done with these cells. It's too slow to have everything be done with human hands. So high throughput characterization done by lab automation, by robot scientist systems, as they're called, and so on. So that's the second thing, and the third thing really is just funding, because one thing that's true in this field is that nobody's stuck. We are not to a point where no one knows what to do. Everybody who works in this field has a lengthy research agenda. We know exactly what we need to do. The roadmap is developing very nicely. Every time you do something, you see 10 more things become possible. And the rate limiting step is funding support, which is extremely precarious in science always. It's even more so today. And just the degree of continued reliable support for the young people that are entering the field to make these things possible is really critical if you want to see the applications bear fruit.
Well, that's amazing. So, Michael, thanks a lot. I think our audience really enjoyed discovering like really this truly new field. And we are as well like looking forward to welcoming your colleague Joshua to the Global Summit in Geneva, 8th to the 11th May, 11th July.
Definitely everybody should catch that. He's an amazing speaker and he's got incredible data. So everybody should catch it.
Yeah, no, that's going to be amazing. So we're very much looking forward to his participation as well in the exhibition area and explain us more about the Xenobots. So thank you, Michael, again, and as well to all the attendees for making it such an interesting session. And we encourage to check out the AI for Good program online to see more robotic sessions that may be of interest to you. And this is the end of this section and the start of a brief networking in the AI for Good neural network with Michael and participants for the next 5-10 minutes. So see you all in a couple of minutes now, and I will give the floor back to Anna for the closing information. Thank you very much.
Yeah, thank you. Thank you, everyone.
Thank you, Michael. Bye.
Thank you for participating in today's AI for Good session. We hope you've learned something new, innovative, and engaging in today's event. We now encourage you to continue the conversation on the live video wall in the neural network. Here you can ask questions, like and comment, share links, complete the poll, connect with interesting profiles, or speak one-on-one using the chat and video function. We invite you to explore the lobby, try the smart matching quiz, visit the virtual exhibits, poster boards, the eShop, and build your personalized AI for Good program. Let's shape the future of AI for Good. you.